Class 1 histone deacetylases, like RpdA, are discussed as potential novel targets for the treatment of fungal infections. However, purified enzyme activities are required for their further characterization.
The main advantage of this protocol is the rapid and sufficient separation of native TAP-tagged complexes for activity determination in one single step.
HDAC complexes derived from this protocol may be used for efficacy screening of novel fungal-specific deacetylase inhibitors.
This method provides the basis for the extraction and purification of TAP-tagged fungal proteins, which might be used as starting point for the establishment of protocols for other enzymes and strains.
To begin this procedure, add 10 milliliters of CSS to each flask of prepared canidia. Tightly close each flask with the provided screw cap and shake vigorously. After this, use a sterile inoculation loop to completely scrape off any remaining canidia. Pass the canidia through 40 micrometer cell strainers placed onto a centrifuge tube and collect the suspension from 5 of the flasks into one tube. Then centrifuge the samples to combine and dilute the samples as outlined in the text protocol. First, place cheesecloth into a funnel on top of a flask. Filter the prepared mycelia through the cloth and wash briefly with deionized water. Remove as much moisture as possible from the mycelia by squeezing the cheesecloth between just hands and then between paper towels.